Hi, I'm Liam Gershoni, and my project is called Does It Mimic? It explores how certain epitopes from viruses and bacteria may be involved in triggering type 1 diabetes by mimicking certain self-autoantigens found in our pancreas called hybrid insulin peptides. The leading theory for that trigger is molecular mimicry, and it works in 3 steps. First, an antigen-presenting cell breaks down a viral protein that happens to look structurally similar to one of our own beta cell proteins. Second, it displays that fragment on an MHC class II molecule and trains a CD4 T cell to treat the shape as a threat. Third, and where the problem stems from, because one of our own proteins may share that shape, those same T cells cross-react, infiltrate the pancreas, and destroy healthy beta cells. The self-target this project investigates here is unusual. It's called a hybrid insulin peptide, or HIP. HIPs form when a fragment of proinsulin fuses to a fragment of another beta cell protein, like chromogranin A. It's a fusion that isn't written in the genome, so the immune system was never trained to tolerate it. In 2019, research showed that HIP6 and HIP11 can activate specific T cell receptor clones on specific HLA molecules. This makes them good candidates for molecular mimicry. If a virus or bacteria carries something shaped like a HIP, it could be a trigger. So how do you search for something shaped like a HIP? First, you have to understand the target. I took 5 TCR peptide MHC complexes that are known to trigger T cells and rebuilt them in AlphaFold 3 so I could see exactly how the HIP sits between the receptor and the MHC. T cell receptors don't read the amino acid sequences like text, they read 3D shape and charge. So a true mimic doesn't need the same letters, but it needs the same physical surface. That's why my search patterns allow conservative substitutions, essentially swapping an amino acid for another one with similar chemistry. I ran those patterns across the proteomes of viruses and bacteria linked to T1D using ScanProSite, and the search identified 1,092 candidate peptides. Each candidate then went through 3 tools. First, NetMHC2Pan to predict how well the peptide actually binds the MHC molecule. Second, TCR Model 2 to build and visualize the full TCR peptide MHC complex and score how well that complex holds together. And third, Prodigy to calculate the binding energetics and thermodynamics of how favorable the interaction really is. I judge those complexes on 4 metrics. 2 come from the structure model, TCR PMHC IPTM, which measured confidence in the binding geometry at the interface, and IPL-DDT, which measured confidence in placing the exact side chains that actually touch. 2 are physical, Gibbs free energy, or delta G, which measures how favorable the binding is thermodynamically, and the dissociation constant KD, which measures how tightly the pair stays bound. The profile of a real mimic is high confidence, low delta G, and low KD. A Welch's t-test compared each candidate's interface confidence against negative controls, peptides that shouldn't bind. Of the 55 candidates that reached this stage, 47 were significantly different from those controls, and because structure prediction is stochastic, I ran each one across 5 random seeds and built 95% confidence intervals on every metric. After that filter, 10 candidates met or beat the native HIP on all thresholds at once. To rank those finalists fairly, I built a composite score, taking each candidate's TCR, PMHC, IPTM, IPL-DDT, and delta G as a percent change from its native HIP, standardizing and averaging. 2 complexes produce statistically significant mimics. The most interesting result comes from a SARS-CoV-2 peptide, which outscored the native hybrid it's imitating. This is significant because clinicians report a rise in new type 1 diabetes cases after the COVID pandemic, and this gives a concrete, testable mechanism explaining that link. I would like to thank James Heather for developing Stitcher, which was a foundational program to this work, as well as for mentoring and supporting me with my research. Thank you.